特別嘅嘉賓speaker 咧，今日來頭真係不少啊！如果我要講咧，就真係可能講多半日都講唔晒。因為Professor Dr. 莫英健教授咧就係首席醫療官喺好多個組織裏邊咧。過去咧，佢早年喺香港大學嘅內外科醫學院學士啦，跟住亦都係咧接受咗呢個腦源神經培訓。我通常好少睇貓子嘅，不過因為咧太多專業詞我都唔敢亂咁講，所以我都要捉到貓子，亦都證明呢個 topic 對我有重要影響，係咪？大家都可以要正視呢個 topic 啦嚇。咁啊，莫英健教授咧早年咧就喺香港亦都完成咗腦源神經嘅培訓啦。喺香港內外科醫學院院士及英國愛丁堡皇家內科醫學院榮授院士。咁佢特別咧由佢嘅研究嘅興趣咧就由呢個臨床神經科啦轉向咗呢個神經遺傳學嘅研究。喺倫敦大學學院博士之後咧，佢亦都繼續鑽研佢呢個喺關於呢個柏金遜啊同埋阿爾茲海默症嘅方面嘅研究。佢而家咧亦都係咧香港神經退行性疾病中心首席醫療官，同埋香港科技大學生命科學部嘅臨床教授，clinical professor。咁佢發表咗多篇文章啦，關於頭先提到嘅柏金遜症啦、阿茲爾海默症啦，同埋腦源神經病。咁頭先噏完一大堆嘅，個字都識係咪？咁拼埋就唔係好識啦，我哋就。咁今日就由Professor Dr. Professor Mo 咧去同大家去講解。今日不過咧，亦都請咗另一位高手出場。你知我哋百川論壇咧就真係臥虎藏龍嘅，高手雲集嘅。咁我亦都今日咧交個咪咧同等Fred Wong 我哋嘅老友記Fred Wong。Fred Wong 咧亦都係eFusion 嘅Chief Investment Officer。咁有乜關係咧？證明咗呢個位置你好有吸引力啦，同你有磁場啦 ，OK。咁佢對腦源神經咧 neuroscience 係非常有興趣，亦都自己有佢嘅興趣之一。對投資因為佢 remind 我咧就唔係講緊 Chief Investment Officer，remind 我咧就係信報專欄咧其中一個咧叫做咧再見瘋人院，其中一個4個病房裏邊其中一個病人，夠晒癲啦！唔癲做唔到投資係咪？所以咧，今日我哋以下落嚟嘅時間咧好精彩，交俾兩位高手，等我哋希望能夠多啲火花。咁亦只要稍後咧，再後嗰 part 咧就會有 Q&A 嘅時間嘅。大家準備定一路今朝早運用我哋嘅腦源神經啦，大家運作起嚟 active 啲。跟住一陣大家踴躍發表問題，睇下 Professor 咧一路同大家解答我哋呢個咁複雜嘅腦源神經，好嘛？睇住掌聲Fred Wong 同埋Professor。
先我偷睇咗少少Professor 嘅PPT， 然後咧就今日有機會可以同大家brief 少少呢個佢嘅內容或者係都好榮幸有機會代表百川學術，即係我係 N 號員工，N 號義工咁唔知第幾個，咁好榮幸啦。我成日都研究呢個腦科學嘅，咁可能之前我哋研究 AI 同我兩個仔喺度講。喺百川度講呢個 AI 嘅時候，都成日提到呢個同腦源神經嘅關係啦。咁今日其實Professor Mo 會講嘅內容咧，當然極度係呢個非常之專業啦。我哋啲即係 part time 同埋係得閒喺度諗下嘅只係嚇。咁但係咧，我哋想有3個 inception point 有3個直入嘅 point， 我覺得咁因為做 moderator 有呢個機會可以講其實我哋琴晚最精彩或者前幾個禮拜最精彩同我哋息息相關嘅係乜嘢咧？其實係啲股市大升啦，係咪？尤其是係韓國股市，美國股市。咁入邊咩股票升得最多咧？其實係 Hynix、Samsung、Micron 係咪升到傻咗？係咪在座冇人揸住呢啲股票啊？有冇人冇揸住呢隻股票啊？全部。咁點解我講呢啲咧？其實即係記憶體啊嘛，係咪？因為我以前都係做半導體晶片設計嘅。記憶體其實係乜嘢咧？就分2種嘅大類嘅，一種就係揮發性嘅記憶體，一種係非揮發性嘅記憶體。即係一種係瞓咗覺之後就冇晒啦，就揮發性嘅；另一種咧係長期記憶。咁我估咧，因為Professor Mo 咧其實係即係 UCL 畢業噶嘛，係呢個權威嚟噶嘛，即係世界級嘅權威，因為係我聽講係繼哈佛之後 UCL 已經即係2大嘅係喺 Dementia 喺呢個 研究呢部分係即係世界級嘅頂尖嘅第一、第二名嘅係我以我嘅粗淺認識應該係咁嘅。
第二、第三啊，第一名科大。
第一名科大。啱，第一名嘅科大。所以海馬體咧呢樣嘢，即係唔知大家知唔知海馬體？海馬體其實就似係短期記憶咯嚇，即係似啫，因為我係門外漢嚟，我唔係咁精準嘅啲字眼。咁但係我覺得海馬體係好似我哋嘅 DRAM 嘅。咁 DRAM 就係而家 HBM 咩高high bandwidth memory 啊嘛 HBM 就係令到呢股票升幾倍嘅原因啦。一年之後升咗幾倍喎，咁所以呢個同我哋好有關係啊。所以聽完之後就知道應該點樣買賣呢個記憶體咯，係咪？不過唔作投資建議啊，當然我幫 Professor 講下呢個 disclaimer。第二個咧就係、其實同我哋每個人都相關嘅原因係咩咧？因為我媽咪有少少似係有dementia， 我外母都有少少似嘅 dementia， 但係佢哋80歲到啦。但係我哋在座個好可能咧，幾年之後我哋嘅 cancer 可能都會被醫到嘅，大部分 cancer 都可以醫到。我哋嘅身體可能可以跨越120歲，甚至乎更多。咁但係當我哋發覺90歲你未有 dementia， 你100歲有冇咧？110歲有冇咧？咁有啲人就話：唔使驚，我哋以後有 Elon Musk 嘅植入晶片啊嘛，係咪？咁所以我哋每個人個腦咧就揀個位置就開個窿咁樣。你想唔想咁咧？所以呢個我哋好可能個身體可以跨越120歲，但係我哋個腦未必跨越到喎，未必可以 naturally 可以跨越到，可能要借 SpaceX 嘅唔知乜嘢啊，借呢個 Neuralink 嘅唔知乜嘢。咁其實係咪真係得咧？同埋如果換咗個 memory， 仲係咪你自己咧？換咗個短期記憶同埋換咗個長期記憶，係咪換啦？即係 partially 去 stimulate 或者 whatever 啦。咁仲係咪你自己咧？咁呢個係有少少哲學上嘅問題，同埋同我哋息息相關嘅，因為我哋每一個人都有機會跨越120歲，可能係極可能係應該係做，我覺得係。咁第三咧就係同我哋有關係嘅嘢就係、我覺得我成日都話點樣可以即係我個人比較鈍啲啦，蠢啲，反應慢啲。咁其實點樣可以令到自己聰明啲咧？咁今日我自己好期待就聽到阿 Professor 嘅演講咧。其實點樣令自己聰明啲嘅最直接嘅方法係咩咧？係研究聰明本身係乜嘢？咁點樣研究聰明本身係乜嘢？其中一個就係做多啲 gym 咯，唔係去24/7啊，唔係去 pure 做 gym 啊，係我哋個腦袋做 gym 咯。咁腦袋點樣做 gym 咧？咁我估啊 Professor 可能會提到呢個 neurogenesis 咯，同埋 neuroplasticity 咯，即係個腦再生啊。腦再生其實就係做 gym 咯，我哋嘅腦袋不停做 gym。咁係咯，我哋用熱烈嘅掌聲可以歡迎啊 Professor 同我哋講下呢個人腦結構，好值得探討嘅題目。我覺得人生冇其他題目正比呢個題目更加值得探討嘅。
好多謝Albert 請我今次嚟嚇，咁係有機會見到咁多新嘅朋友，咁希望以下落嚟會有一個好嘅交流嚇。咁或者先講少少就係，因為最初係有即係在座咁多位係來自各行各業嘅精英，當然有學生啦，即係我哋嘅未來啦。咁因為係咁，所以我今日諗嘅題目嘅時候講或者講嘅方向咧，係冇諗住作為一個譬如話同學生講，又或者同醫生分享嘅，即係我唔會用嗰個角度咁精準咁。其實我今日講嘅嘢大部分係我嘅興趣嘅嘢，但係甚至有啲嘢根本我唔係好熟悉嘅，即係話我之前都提過今日 Alzheimer comfort zone 嘅嘢係最多。即係，但係呢個全部都係同遺傳同埋腦有關。咁我希望可以借住呢個機會，大家聽完之後覺得，咦呢樣嘢我有興趣，咁作為一個大家嘅分享，同埋我好着重覺得極重要嘅就係唔同嘅 professional， 即係唔同嘅界別裏邊跨界別嘅合作或者即係刺激或者腦震盪。咁因為我哋嘅進步，其實我哋嘅進步唔應該係喺一個細嘅範圍，即係其實原來你都唔知原來隔離嗰個做緊咩我哋個進步其實最大重要性就係即係 你從歷史上都睇到嘅，唔係單係自己嗰方面係將好多嘢整合埋。Because I was told that there will be someone that is not that good in Cantonese as well as some non-Cantonese, non-Chinese speaking. So I was just to give the talk in English. But I'm always happy to answer or change a little bit into Cantonese, which is in fact my most favorite way of chatting. Sorry. So this is just my 5 main categories of, subtopics that I'm going. So I'm focusing, because I want to talk about a little bit every different aspects. So I divided that into different smaller topics. But before that, as I said, it is about genetics. So there are 2 slides that purely talk about genetics in general. First, I believe you don't necessarily to believe. I believe that a lot of the things is coded in our DNA, and our DNA, well, you can say the length is 3 billion in terms of each and every bit. So which is huge, and you can count from the very beginning to the end. And so when we say that, okay, how the DNA affects us, it is the DNA is in fact first get into we call it transcribe to RNA, and then first of all, the DNA is the blueprint, and then it will be transcribed to the first product, that is RNA. And this RNA in fact goes through 2 steps. First is an exact replicate of the DNA. But then these are, some of them are, well, you can say useless, or you can say it gives another way of doing some modifications. And then it truncated, that is cut into another shorter mRNA, which in fact this one is coding for the protein. And then when the protein, when it is used for coding the protein, in fact when they code it is not that they transcribe a single chain of amino acid and then change, but in fact it is when it transcribe it, when it start to translate, sorry I used the wrong word, when it translate it already start to fold. So each of the steps give you some variations, and each of the steps give you modifications, give you some way of manipulating. So if you go from computer, or you have a lot of more dimensions that is outside, well, when you want to calculate or you want to put in a model, this is something that the factors that you need to take into. So the next thing is genome-wide association studies, because a lot of what I later present on is related to this topic is, well, it is association because when the results don't give you a cause effect answer. That is, when you find something that is associated, you are just you are just, let's say they are linked together, but they may not be affecting. So one may not be the cause, it can be just happens to be always together. Let's say, well, you see, a lot of people are always happens together. They are just related. They may not be family. They may not be. They may not be even close friends. So linked together is just association. Cause is another effect. But this is one way of you can do. Is because it allows you to study a large populations, get a lot of potential questions without first assuming, this is probably A causing B, so I'm going to study it. How it is going to do it is in fact two groups of people, so-called case or controls, and then you will be looking into a particular bits of information over the 3 billion chain. So in 20 years ago, it is talking about tens of thousands bits. Now you can talk about the whole 3 billion, roughly. So let's say bits 101, then we study how many percent in case, how many percent in controls. Okay, there's a statistically significant difference, then we say, okay, this is likely to be some sort of association. And then the results will then link up together, give us a plot, which we always call, nowadays we call it Manhattan plot, because when we plot together, assume if this one has now typically it's about 5 million dots to form this particular plot, then there are some that is particularly high, and they say, oh, it looks like a Manhattan of the New York City of this skyscraper. So that's why it's called Manhattan plot. So these are concepts that I would like first to talk about before, so that everyone's understand, and then we talk about some later on. When I give talk, particularly not very formal one, I always like to quote something from the past. 誰故之初，誰傳道之. It is one of the very old questions, because the whole manuscript is about questions about how it comes, what happens in the history in the past. I guess what you did not mention about his ancestor, because he did mention where he come from in that particular sense. So when we ask about where we come from, the popular thinking is we come out from millions years ago. Not we, if you, depends on how you define. What comes out from Africa is probably a humanoid that is closely related to humans, or to be more specific, Homo sapiens, that is our species, but it is a little bit of the ancestors. And so there are different lineage. So this part is my always interested appetizer, because this is genetics, this is about us, this is about our history. Okay, there was no other picture of a human except this guy. This is Dr. Fu Haomui. Well, she's of course much younger than me, but she's one of the persons that I admire, because for the students who do in research, these persons have yearly paper in Nature, in Science, in Cell separately. It's not either one in the last few years. So she is one of the most important persons in the field of doing genetics in the archaeological samples. And that she tells us who we are in terms of we Chinese, where we are coming from. So just to look at this, the color one are those coming from the genetics of the archaeological samples from different places, and the other background are the current genetics from the Australians up to here in the Asia, in the Mongolia, and some this is also Australasia. So compare with what is taken out from those places that is shown, and for those, they usually divided into north and south, and for this particular slide, just look at the color. So this is the northern part of the East Asia, this is the southern part, this is the time frame that is the oldest, that is on your right hand side is the newest one. So the color of the oldest, not that old, is just 10,000 years ago or something around that. They are all Homo sapiens now. They are ancestor and human, they are not hominoids. And then about the other is about 5,000 years ago. And so you can see from the much older time, the north and south looks a little bit, well, at least you can see the north is green, the south is a little bit on the blue side. At the present days, it is more green. That is, a lot of our populations, we Chinese, are coming from the north. Well, it could be after the ice glacier age, or at the end of the ice glacier, at peak, before it gets warmer, they are forced to go downwards. Could be. It doesn't give an answer, but at least first, a lot of us are coming from the, originally more from the north. And then the other things is, you can see from more recently, compared with those different type of populations, this one is coming from Vietnam, old Vietnam ancestors. For the most recent one, you can see from the Southeast Asia, a heavy population, a heavy loading is coming from northern China. So it seems it is going from up there down. But for the Chinese, current Chinese, it's more likely to be coming from the north. But, well, later on, we'll see that there is evidence that not only north, but we also have southern elements though. So we are a family, we are mixed all within coming from China. So this Sacmao is one of the very large archaeological sites in Chinese that is well preserved. Dr. Fu also participated in using those samples. In fact, you can see in this burial grave, by using those genetic studies, they link up these, well, who in that particular noble courtyard. Three things that I would like to mention about that is, first, in those genetics, compared with the north-south distributions, there was south. This one is in the Scinece, which is pretty north. So there was, even 5,000 or 4 years ago, there was some effects that comes from the south up to the north. So that's why I say we are mixed. Although there may be a dominant genetics coming from the north. The second is there's rice found there. So it's not the work of Dr. Fu. It is really the professional archaeologists that found remains of rice. So that means that, okay, again, rice was originated from south. So that springs up there. And also, one minor point is, well, of course, in those days, we bury human alive, or may not be alive, but at least we bury them for sacrifice. And the archaeologists think that, oh, most of those seems to be females. But Dr. Fu said, oh, genetically they are male. So kind of it's a side track that this is the importance of multidiscipline. Without these new genetic techniques, Dr. Fu won't be able to tell a lot of these things. You can just only read from the documents, or you can only read from what is taken out from the ground that, oh, there seems to be close, oh, there's a rice grain here, so probably it comes from south, but is it really they are using it, or they are, but then if you find some people that it has genetically, in fact, it's coming from the south, so, oh, there's interactions they bring along, which is more supportive. So just jumping quickly is Yunnan, Wannan. It's in fact a site of interactions. A lot of the genetics come from our north and southern China, going to there and then interact and then spread out. It's in fact a important crossroad. Also, just by coming looking from the genetics. And then here you can see again, talking about genetics interactions. A lot of them are okay. Previously, about 10,000 years ago, the southern Chinese interacting in the Yunnan and then going further south in the Southeast Asia. And then, but however, a lot of the northern genetics also shown up here is probably not that many years ago. Okay. Interesting part, around 1,500 years. Anyone in history, Chinese history? That part is 魏晉南北朝，衣冠南渡. And for this one, what is the rough age of that? That will be just before Ha Qiu or around Ha Qiu. So is that the legendary Wang Dai? Some people say that, but I mean, these are the interesting part, but these are all coming from archaeology and genetics. So it's almost end. I always would like to have some interesting things. Dogs also migrate. Migrate is in fact genuine. Is Tonga is in the southern Pacific, highly remote. Before the dogs that are there before the Western people comes in, if you count the genetics, it is highly related to Saigon, Zhonghua Qi. So here is what genetics can tell. I mean, is it related to cognitions? Yes, I would say if we are not not evolving from a homologs to Homo sapiens to human, with our brain improved, with our social life, we won't be able to talk about cognitions. So I'm not going into detail into it. It also supports because of this is another 交通要道 or crossroad, the Mongolian plateau regions, and in fact there are some exchange in the different types of people, and one of them affects Hongshan Manfa, which is very important early culture of our ancestors. So they're now more hardcore science. Well, not that hardcore, but it's no longer archaeology or other well, archaeology is also science, I would definitely say, but it's now more biomedical science. So of course, I mean, we differ from a lot of others, from non-primates, from mammals that we use in the lab, say the mice. So you can see, well, although the structure, it is to the scale from this particular paper they specifically mentions. It is size-wise is up its scale. So you can see we have a much larger brain, even for one particular part. This is the cortical thickness of the brain cell part. You can see we are much thicker and we have much more complicated. So compared with those more primitive animals, and even within the homologs, that is human-like but not human, compared with the current day chimpanzees and compared with the modern day chimpanzees, you can see that the cells, the dark dots, is in fact reflect some sort of connections. And when you compare with the human one cell has much more connections, which means that it is more complex, and which means that it is more difficult to model after. And in fact, I would say with due respect to the tech side, the tech people, 20 years ago, or maybe some sort of 15 years ago, in European, they have a project. They want to have the computer to model the whole I can't say whether it's human, but I guess it is some sort of thing. It is not a small funding. It talks about tens of billions euros. And in the past, it started. But now, you don't hear much about that because you see, we have our cells in the brain are probably more than the stars in the universe. I'm not sure that is correct, but you can think of so many cells. If each have a couple of hundreds or thousand connections, what will be the number of permutations or possibilities? And then some people, I guess there are those that did not discuss with the biologists before they get the grant. So which means that you can't simply model a animal or human-like brain without getting some sort of reductions or assumptions. So of course, a lot of our knowledge comes from embryology. So that is human embryology. You see how it develops. Well, at the very initial stage, if you believe, which is, well, at least morphologically, it looks like lower types of animals, the fish, a chicken. At the very early stage, the embryos look similar. It is only later on that the brain grows, enlarge, and then form our postnatal brain-like. And within the cells, then you can see there are much more complicated development. And Mr. Wong just mentioned about neuroplasticities, the growth. Interestingly, I don't know why it is like this. The brain cells, it grows from the downward side is near the ventricles, that is those that the fluid side, the others is the outside of the brain. When they grow, it grows from here, divided, and then climb all the ways. So the outer one are in fact pretty later developed, which means that, well, when we develop our brains, not only it need to divide, it need to climb, it need to find a way to the appropriate place, and it has a certain distance to go through, which means that it is a complicated procedure, how they navigate upwards in the correct positions. Because if you don't navigate correctly, if you have a cluster of cells, then it will be abnormal. Some of those abnormalities, let's say some congenital epilepsies, can be related to these types of developments. So, I mean, I say we learn a lot initially coming from the embryology, but you can't keep on repeating embryology because there are not that many embryos that is you can use, even discounted the ethics, because I mean some people may not allow human embryos to be used to study, but at least if you are allowed those aborted embryos to be studied, you don't have so many. So with technical advance, then of course we have the brain inside a dish, which is we take out some of those cells, human cells. It can be even from the blood, and then we reprogram it, which gives the Japanese professor the Nobel Prize in reprogramming into a cell that can further develop or differentiate it into neural cells like. So it can be that's why I say in a dish, this is small plastic plates that you put in culture disc, and then it develop into neurons. You can study this. A newer technology, but much more expensive, I would say, is, well, you heard about the mini brains, but well, it's a good gimmick to call it mini brains, but it is far from a brain, of course. But we call organoids. That is, we let the cell grow three-dimensionally, forming a little bit. I have a picture in the next slide. That then you may able to differentiate into certain more like a particular part of the brain, or it can develop into a particular organ, and you can use it, or even to implant into an animal for further studies. It can be coming from a normal person, and it can also be coming from a person that has a particular genetic disease. That is, you know that there are some abnormalities there. So the on the left-hand side is an organoid that tries to mimic a eye, differentiated to that stage. So the two dark ones are the eye pigments. But again, can you say it is a brain? Of course not. I mean, but it is always a good to say, oh, look at this is a mini brain. This is a cross-section, not from this one, of course, but another picture of that. The red ones are some of the highly differentiated areas. So, using these new techniques that we can try to do a lot of other studies. What studies can we be able to learn from using this? Well, we can, as I mentioned, if we manipulate the genetics, then we can know, okay, if this gene is mutated, how it will affect the development. So you will be more, give us more knowledge about how the brain is developed at the very initial stage. Give you some hypothesis so that you can test out in other situations. So I will see this as a more hypothesis generating way of doing things so that you, this gene is known to cause some, let's say, abnormalities in the childhood development conditions or maybe ADHD, and then we put it here and see how it affects the developments, or maybe even use it to differentiate, for some sort of preliminary studies of the potentials alleviations. The other is, okay, also use this to do some of those environmentals. In this particular one, they say it's Zika virus. That is originally a couple of years or maybe a decade ago in South America, those mothers that have infected with Zika virus, the fetus is born with a much stronger brain because that once affected, the development is impaired. So you can put it into an in vitro, that is in a dish environment outside the really non-human situations to do the studies. And of course, we can put a lot of those, these are some syndromes or disease that we know that can affect our developments or early stage of the developments. So in helping into, let's say, using those in vitro studies. So I try to do everything that is interconnected when I do the archaeology part as my appetizer. In fact, there was some meanings is that, well, how we do this is using the current human data around the whole world. I mean, if between different populations and if there was a genetic difference, we can try to estimate, okay, what that particular genetic difference, how early it is coming from developed. So this is the way is they use a worldwide populations doing some sort of the genetics scanning and then calculate each and every bit that is known to be different and to chart it, okay, what is the estimated development time before now, from 1 million or 2 million years here to the most current. And then they correlate that with those variations or mutations that is found to be causing disease. And what is interesting, first, of course, it kind of, you see different peaks, which may be correlated with getting out of Africa, recent development. And when you consider those that are associated with phenotypes, again, some interesting findings which I'll talk about in the next slides. And of course, if it is rare, it tends to be occurred recently. So these are coming from known variations that is associated with problems associated with disease. And the interesting findings is, well, the neurocognitive one comes from about 500,000 years ago, morphology. This is a time when there was a lot of, from humanoid to human development. So again, what I said, correlate with what we collected from other information about how the human change or how the human develops. And interestingly, some of the most recent changes that if you chart it where these mutations of genes are most commonly associated with the brain, it is some sort of related to our language. And of course, alcoholism-related traits is one of the most recent. I mean, when you're a hunter-gatherer, you don't collect enough grains to get wine, or you don't collect enough berries to produce wine. So there's no selections on those symptoms. So it's an interesting part is it correlates with all our, in a way, roughly correlated with our development. This is somewhere genetics also tells us about our early current human brain development and cognitive development. So a little bit meat now, that is disease of the current day. So I choose two, one is at the early stage, one is at the later stage. ADHD, so 多動症. It's common, it's so common. Well, just then we talk about, I can't remember who we are talking, I have the conversations. Previously they say there was not that many, but again, this is a complex disease. How frequent it depends also on how rigorously you want to look into and to see how whether they have or they don't have. So surely, the coming two slides, there's one main message. It is definitely some genetic related, but I cannot tell you more than that. Because, well, of course, it is a complex disease. Complex means complicated. There are lots of factors, there are lots of other things. So it is difficult sometimes to, from associations, to say that, hey, this is really related. So sometimes it is difficult. And it is only recently pulling a lot of data, which is in terms of tens of thousands of data, that they kind of, okay, genetically there seems to be definite evidence it is related, and not just common and also rare variants, which means if you correlate with the last few slides ago, the rare are those developed later on, most recent. Likely the common one are those earlier ones. So both these early days and also the common recent days ones are related. And we did not yet go into some difficult ways of doing genetic studies, because what I always mentioned previously is just only 1 point mutations or 1 point change. But in our genetics, there are in fact common, and there are definitely some situations that you have a small chunk, or occasionally you have a large chunk that is removed or additional, which is difficult in the previous technique to develop, to detect, sorry. So sometimes you need to have a prolonged studies, you need to have a large number, or you have to have extra techniques. And further confirmations that genetically is related to ADHD. And the point that I want to show is, in UK, which is I think in that, because they do have a public health system for quite a long time, and they do have a research settings that is already in place for quite a long time. They have a lot of cohorts, which is selectively coming from a certain, maybe randomly selected, or maybe from a region that they collect and then follow up for a long time. There is a 1950, if I remember, or 1956, which is those people that born in 1956, and they follow up a long time. These are the most difficult ones to collect, because it's easy to collect them at the particular point of time once, but then if you are to collect them multiple times along the time, it is difficult. Just like cheese, if you have cheese that is old, that store longer, is expensive. The other is control. Italian, if you are talking about the Italian, a 50 years versus a century control, it is 10 times the difference, if not 100 times of difference in price. So longitudinal studies is difficult to collect and it gives us a lot of information. Let's say, talking about longitudinal studies, one of the studies, not large, it is in America, is from a particular order in the Catholic Church that the nuns, just a couple of hundreds, they follow yearly detailed examinations, yearly data collections, and then at the end of the day, they are happy to donate their brains. And it's just only a few hundreds of them, and they give proliferative information that we frequently refer to, even nowadays. So related to this cohorts, I would like to show you, in fact, when you collect cohorts, it is not, you name a person, you see, hey, 5 years later, just a phone call, hi, Mr. X, are you okay? This is not good enough. We need to have multiple parameters. And in this Beijing Institute, they, again, looking into development, early development in the childhood. They really standardize everything, the collections, the scales, standardized way of doing an MRI brain scan, standardized way of doing the EEG, how to read it, how to store it. These are the fine details, but this is the difficult part that gives you important information. And at the back end, the lab, which is, this is the things that I try to want to show, and I hope it can.
机器人不能太快，所以整个流程是加速的。然后那个就相当于他把空管子拿过来去自动化的开盖，这就是标准的动存管。然后这是全国过来的采血管，他就在这儿把采血管里面血分到那个动存管里面，然后就可以自动的把那个动存管盖子盖上。这中间所有的信息都是串联起来的，然后分好的这个动存管里面有血液，就由这个刚才那个机器人就运到生物样本库这边。然后就存进去，然后产线空的时候，机器人就把里面的样本运出来，在这边有个传递的窗口，它把它放到窗口里面。这个屋里也有一个机器人，这个屋里的机器人就把那个样本取出来，放到这个环岛上。进了这个环岛，后面就都自动化了。就是其实现在这些操作都很简单了，无非也就是破碎。然后选取特定的片段，加接头、扩增，然后用磁库就可以复制，然后建完库以后就测序，然后数据下机。然后那个就是样本库那边其实
Standardization, and standardization. When we do our lab findings, we know a lot of biological samples, how you from the point that you take it out from, let's say from the vein, that the blood that is drawn, how it process have an important impact on the testing results. And so this is, if you are doing a lot of, of course, I'm not saying that human is not good. The best artisans will be maybe the best, but then the best artisans may not process a lot of samples. So this way is, they start from the very beginning when they define how you transport, and then once you transport, you put it into there, it in fact controlled, almost automatically standardized way of preparing, extracting the DNA, extracting the plasma plasma from the blood, and do all the, let's say the biochemistries, the biomarkers and stuff. And so, well, so automatic ones is not available now in Hong Kong, but we look forward to in future if there is a chance. So furthermore meet the topic cognitions. What is cognitions? So cognitions, well, it is in fact a philosophical topic, but definitely cognition is not a single thing. That is not, well, at least I believe. There's nothing about one thing about intelligence. One thing, intelligence is not a single thing. Let me quote interesting samples, and for the medical students or medically related, there was a well-quoted case, an American farmer that had a very minor stroke. After the stroke, he couldn't recognize the face of his family. Well, poor enough. I mean, he can't differentiate his doctor, his wife, or well. But when he talked about his friends, about that particular occupation, I mean, he is up running about you. It doesn't look like a post-stroke patient. And when he talk about his farm, he can name the cow by the picture. That is, he recognized his own cows differentially. So recognizing the face, not the face, the color or the shape of a cow is different from recognizing human. So at least this part, just recognizing is different. So this is not one single process. Calculations, arithmetic is one thing, control is one thing. Let's say I want to press this button to do the next slide. It's different from my way of talking, the language, and we have not even talked about what is memory. Is there a physical substance? Of course, if there was a heavily devoted religious people, they may think it is spirit, it rests in heart or in brain, whatever. Well, I try to be more materialistic. Is there a substance? Or what is the physical base? The word engram, to me, is just a term. It did not clarify what exactly is this particular, the memory trace. So don't take this word importantly, except use it as a search name. So some technical parts. Before we go to that, well, genetics, optogenetics is perhaps in the last 2 decades, one of the very important techniques in doing studies in neurobiology. You can implant some particular proteins through various ways into, let's say, a lab animal, and these proteins within the cells, if you shine a light of a particular wavelength, and then they can excite those proteins. This will be either you can manipulate to be stimulating the cell to fire, the brain cell to fire, or inhibiting brain cell to fire. So this is using lights, you can cause it fire or inhibitions. There are a lot more details, but of course, I list all those references. Anyone that's interested in future can go to that papers. So using this to use studies about memory. In this particular situations, it is using mice. So baseline, doing all the preparations, and then doing an electric shock, how to see how it memorizes what exactly those cells are. Of course, you try to you have to label it with some sort of tagging so that you know where the cells are. Then if it is in natural situations, they will produce natural reactions. But if you shine a particular light, wavelength of light, you can, without those conditions, reproduce. Because in mice, in shocking, they try to freeze, they stop moving. But then if you shine the light, even without the shock, they will be doing the same. So they kind of, okay, these cells are related to this particular bit of memorizing that, shock, and then I will be freeze. Reacted in freezing way. So this is the interesting part is, you can see, it is this physical base of memory is one of my weakest parts, so I try my best to give my correct interpretations. But if there are some researchers that is in this area, then forgive me if or remind me if I'm talking something wrong. So for these particular types of things, then some previously studies in lab animals, they will see that, okay, in a particular environment, the lights up will be in one thing, just like our padlock, those mama saw. And in another environment, it will give you another number. So kind of it gives you a certain memory related to a particular few positions, particular few cells. But there are people saying that, that outside the cells, outside these few cells, they may also have interactions, which is true, but what is the evidence? There are also the chemicals inside the cells, let's say the RNA. There are also evidence. Let's say some people, if I remember correctly, use those RNAs of a trained mice and then transplant to another mice. It seems to be learned faster. So is that RNA also related? These are a lot of arguments whether it is related. So this is a more schematic, better way of diagram. Before learning and then during learning, okay, these few things are highlighted. If you kind of reinforcement, let's say a manuscript, recite a few more times, the linkage may be even stronger. And you recall, then you retrieve, that is recall, then these big cells light up. But sometimes you may be forgetting, it might be those linkage are weakened, or they maybe have some cells that is degenerated. Genetics also may play a role, definitely play a role, but what role? This is the epigenetics, that is from the external environment, how the chromosomes is manipulated is also related. But again, what is memory? There's no answer here. No answer here is this is pretty almost a lot of the things are in the last couple of years from others. So this is up to this bit that I confidently tell, but there might be definitely a lot more that I am not confident enough to understand and to tell here. So when we talk about cognitions, neurocognitions, or memory things, what is memory is not answered. So you see how we are far away from answering a lot of questions. And mainly for biological or medically related memory. Well, we think that that I will talk about a little bit more here is philosophical but important, challenging idea, because I don't want to be just like a science. Memory, there are two things, well, at least two things. One is one is semantic memory, one is episodic memory. So I always want to illustrate this, but my memory may fail me. So I may be telling a lie, but that is all I want to say in this way. Everyone knows what is a tiger. So an animal, a shape like that. This is knowledge, this is semantic memory. Everyone should have similar concept, similar idea. Episodic memory, let's say in my situation. My first encounter of tiger is when I was a kid of this height, going to Lai Yun, trying to poke a finger into the cage, stopped by the window. Okay, that is episodic memory. That is tiger is one of the concepts, but then this particular episode is episodic and is individual. So each one have their own episodic memory. Someone may have an episodic memory of their 31st anniversary, forgetting to book a table and then. So that particular bit, no one can tells you how to replace it. You cannot relearn it because this is only you that you know I quote another incident which is real, that when I was in London, a potential PhD student want to apply. Me and my supervisor interview, and then he wants to do, I want to learn about iPSC cells so that I may grow the cells and in future do the regenerations. Which is true, which is fine, which is absolutely, but he did make another even better, he received another even better offer. So we were not able to work together. But after that, I did tell him, I mean, have you ever thought of, if you lose you say Alzheimer's disease, you want to do regenerations to help the memory. Okay, if the cells is already died, yes, you rebuild the cells. But where is that particular memory? Does that help? So a lot of neurodegenerative disease, if that involve memory, if that involve conditions, preventions is almost always things that you need to think of. Not just think of, I believe in iPSC, I believe in regenerations. There are lots of things that rehab plays a role. Rehab can help you to relearn thematic, rehab can able to help you to control the body, to relearn from walking after a stroke or after some other things. But there are things that is not irreplaceable. And we still don't know where it sits. So that is the issue. So any fictions, scientific fictions that at the end, machine tucked into the human brain and then all the memories flow to the RAM or to the hard disk and he exists, he survived, is, what do you want to know? We don't know yet. Okay, two-thirds pass, I guess. So the next thing is aging and chronological aging. Well, aging, we all know our age, but that is chronological age. But across the populations, well, everyone knows, she looks just 50s. So at least appearance-wise, it's not that chronological age, it's just maybe just 50s. So we need to differentiate about, well, in terms of aging, what exactly we mean by aging and what exactly are the biological or exact, or put it in more layman terms, how fit it is. I mean, this particular paper are lots of very senior authors grouping together, giving an opinion about the futures in terms of, geromedicines, geriatrics, that is related to old people, the medicine related to old people. Based on a lot of the already known propagations or known informations, there may be a lot of factors, so they put in a primary hallmarks, which is maybe standalone. Let's say the genetic issues, the telomerase attributions, which is what Mr. Wong talked about, 120 years old lifespan. Some people attribute that length and use that to calculate, it is roughly about maximum you can go up to 120. But well, for that particular length, whether it is reflects what is happening or whether it is the cause, again, I cannot go into details, but what I want to say is there are a number of factors that is well known, that is correlated, or I won't say cause, but at least correlated to aging, to poor, less well-functioned body. And then there are a few things that is already known, that if you have these, it will all in general causing, earlier aging in all aspects. It's regulated, I think it is a very complicated way of saying diabetic and related issues, which is nutrient dysregulated sensing. Mitochondria is well known in all sorts of degenerative stuff, but these are not isolated topics. These are definitely interrelated. Let's say genetics and the diabetes are surely correlated. Genetics, mitochondria correlated. And more functional abnormal protein, well, which one of the cause probability of related to AD is probably again genetic and the other things. So these are the things that they highly correlated, but these are something that we need to think of. Okay, when we want to have a better way of future healthier aging, there are multiple parameters that we can think of. And of course, they then link up what are age-related disease, which means that if you are older, you tend to have lots of more these things. Again, how to correlate these two things together? I mean, the original 14 factors correlating with multiple disease is huge, it's difficult, which we will talk about, slightly touch on. So there are at least some unified single gene that may be correlated with multiple things. So that is, they notice that there might be some genes that is affecting one, but there are some genes that may be affecting multiple factors. And try to do well, first, as I mentioned, in future, if you want to a much more healthier aging, first, you need to think of, okay, what proactive measures, that is, when you are young, or when you are, well, not that young, but middle age, you start off thinking of healthy thinking, proactively doing the right things to help to decrease those pro-aging factors. And then if you are able to screen, then you try to avoid or try to alleviate. And of course, let's say Pigunangfa or other neoplasms, we try to do it as early as possible. But I mean, those are a lot of things already we are known. But to be more precise, then this is the difficult part, because this have to have many factors, many inputs, and how we analyze it is difficult, because that is really the exact part of the big data. You can't do this investigation, that study, or through just only 10 or 10,000. It probably is really a big data, which is again go back to just we mentioned about the cohort. If you can build up a large cohort, standardize a lot of the assessments, and then in future looking into it, correlating back how best we can use it to help. So, quick point about determining a biological age. So there are already starting to think of, okay, we take blood, correlate the markers. This time it's not genetics, but biologicals, compared with known deterioration in certain organisms, organs, and then correlate those markers that may be abnormal, more likely abnormal, then give you a profile, and then kind of individual organs can be give a so-called biological age at this particular point of time. It is a starting point, it is not the end. It is a starting point to study only, because we don't have large scale these types of data yet. But if we are able to build, then we can kind of track how the aging is, and is there any way that we can decrease the biological aging despite the chronological age is increased. So coming to the last bit, if anyone is coming here mainly to listen for Alzheimer's disease, I'm sorry, this is only a few slides and this is the last bit. Again, it's just genetics, it's not talking about Alzheimer's per se. I want to use a complicated, use it to illustrate I say a complex disease or a complicated disease. Is it genetically related? Yes, 100% yes, because this is proven beyond doubt in a way. First, there are families that if you have, there are so-called dominant genetics, that is, if you get one of them, you'll be surely having a Alzheimer's disease. Let's say a certain type of APP mutations or PSEN mutations. But as my mentor in UCL days always told me, oh, I'm not the one that discovered that particular gene APP. It is another one that used Down syndromes to discover that Down syndromes, 唐氏综合症, is a duplication of the chromosome 21, and because of this, the APP loading is much higher. And in fact, the Down syndromes, some may able to escape from Alzheimer's, but most in the Middle Ages already demented, suffering from Alzheimer's. So genetics, yes, but most of the genes are not that particularly significant. A lot of the genes, in fact, I think now it's almost up to 100 regions or foci that is related to Alzheimer's disease, and it is kind of, if you have more, then probably you will be more likely to have the Alzheimer's disease. So a disease that can be caused by different types of genes with different effects. This is what I want to show, a balance. It is kind of really some sort of conceptually, you can see it as a loading. If you pass that particular loading, then you have the disease. It can be caused by one single stone, that is, let's say a Down syndrome, or a dominant APP gene mutations. But you can also be caused by multiple genes that each have a small effect, that is, 將粒石仔將 a lot of small stones, small pebbles added together. And this particular issues of many genes, but each with a low effect, give rise to a what mathematically or scientifically we say normal distributions. Why? Despite it is genetic, because you can see, for those interested can go to that particular website, the Wikipedia website that show interesting short videos. That this is a mimic of assuming you have 4 genes. So which means that because you have 4 genes, it is paired that you have 8 factors that can cause, it will be already distributed in 9 different columns. So assume if you have 100, which means that the distribution is so normal in a way, so normal distributed that you find it difficult to pin down definitely, oh, this is genetic related. So a lot of disease, ADHD, Doppelganger, definitely is a complex disease. It contributes by multiple small genes. It may be contribute a few copy number variants which I mentioned, or there might be some dominant mutations. Whereas I'm not working in that area, I can't remember whether there is or there isn't. But it's a complex disease, and so dissecting the genetic for individual case is difficult. So what I see is that genetics, other than those monogenic, that is the dominant things. In fact, what genetics can tell us is, in fact, try to find a pathway. Okay, these are the genes related. Can I lead to some sort of thinking or pathway? Which, yes, you can see genes. When you put all the genes that is listed together, you can see they group into different pathways. First is the formations of the amyloid in those genes. Some are causing the blood-brain barrier clearance. Some are related to microglia, which is inflammatory related type. Note that I want to point here is ApoE. It occurs in more than one situation. This is another dimension of difficulties, is one gene can be acting on different pathways. So, which means that for a complex disease, at the end of the day, you have multiple factors that you need to factor in if you want to study. So that's why it's difficult to model, and you need to really try the best to reduce it to a certain number of dimensions that you can handle. Otherwise, it's just an observation of the whole world populations, and then one day, maybe with quantum computer, we may be able to do this. But nowadays, although we talk about big data, we need to think carefully what to be input into those data because they are huge. And I probably will skip this because this is just a way of calculating, or you just say how I sum up the small stones, what is the exact, how the weight of the small stones added together, if you want to put it a very layman way, which it always shows some correlations, but just by calculating this, you can't tell whether they will be having, because at best it is 60, 70%, maybe up to 80%. So it's, and at the end I always say, what have I left out? I didn't talk, especially for professionals, because I want to let them know, okay, these are the things that I didn't talk about. But for this particular bit, I will say loads of it. For those who work on Alzheimer's disease, this is not late onset Alzheimer's disease. It's just meaning a lot of things I did not talk about. And I will end here by quoting 莊子，人生也有為，自有無為，以有為佢為可以。So there are a lot of things I surely cannot handle all, but I just try my best.
Thanks enough for professors enlightening. I see most of you are sweating in your brain already, right? So your dementia already delayed for 10 years, 5 years maybe. And after reviewing our video record, you will extend another 5 years. So you delayed the dementia. Yeah. So we can check your brain age later on, right? The brain age, the biological brain age. Yeah. So. Professor mentioned that the episodic memory. Anyone do not understand what means episodic memory? This is very important, I think. Semantic memory and episodic memory. It recorded me the romantic moment when I first time hold my girlfriend's puppy loves girlfriend's hand. That is episodic memory. So emotion and feeling may be one of the key elements for the episodic memory. For semantic, we usually in a logical way. The comparison and break into small pieces, right? So maybe we open the floor for Q&A, the most exciting part. Anyone would like to ask the first question?
Professor, a very interesting talk, and I was particularly interested in you bringing out the optogenetics because 35 years ago I was one of the few people in making excitatory amino acid injections in the brain to identify neural substrates, functional neural substrates. Anyway, there are many interesting questions, but specifically, I want to ask you your thoughts about the idea that, some people think that the human brain is actually, or the entire human organism, function as a data storage, as you mentioned before, because the DNA is actually a coding mechanism. So the function, when it functions as when our brain and our body function as a data storage system, as well as an application. So the information is encoded in the DNA, and the app executes the program based on the genetic instruction. And this was highlighted by your mentioning about transferring of the learner's RNA into another person will facilitate the memory learning. So what's your thought about this?
Because it is not good as far as I remember. Of course, it's lab animal experiments. And I could not go into details about that particular experiments in the publications, so I'm not in a good position to comment. But I do believe that there are a group of researchers that is really working in having a chemical part as part of the important portions of storing the memory. This I kind of believe, but I'm sorry to say that it's outside my comfort zone, it's outside my capability to answer. But I think that will be, well, a contributing factor. But I cannot give you anything. Sorry.
It's okay. Yeah, well, so the idea was basically we were created by aliens or the gods. So they use us as a data storage. And
that is a philosophical question.
Yeah.
Yeah, whether this is the user as a storage of some sort of data is, I guess, is a philosophical question. I'll quote 而家講敬鬼神而遠之 so I'm not going to answer that.
Yeah, Professor, my question is autism. Is that a genetic, cell, or psychologically related or brain-related issue?
And I can almost 100% say that it is partly genetic related because that is pretty well confirmed in a way, just like, in fact, it is pretty kind of the spectrum of the ADHD. In terms of psychiatrics, they are grouped in kind of that particular area. So, but then, as I mentioned, both the two examples I quote for the young ADHD or maybe autism, this is complex disease that will be definitely environmental factors, that there will be any factors that may be affecting through the learning, but there will be genetic factors. For cell, I am not sure, I did not go into details about the individual cell studies that is related in this aspect, but surely, just conceptually, I think if you think of the DNA and you think of the brain, the result is always a summation of the cells inside the brain. So unless you believe in the spirit that is hiding somewhere else, otherwise there must be some physical substance and that should be part of the cells that is involved.
Thank you.
Professor, may I speak in Cantonese? It's easier for me. Okay. 其實咧，香港人咧應該而家係平均歲數係85，個 standard deviation 係15，即係好似由開始咁70歲開始到100歲咧就係我哋叫高峰期啦啫。咁啊最惡劣一點咧就係85歲咧，大概係如果我冇記錯嘅話，一半人有點樣做31。咁都幾恐怖，就算30歲都咁，所以其實我聽到會活到120歲咧，我好驚，因為80歲已經係1/3啦，我諗90歲或者95歲可能就超過一半，咁你冇晒個生命質素㗎，如果你係點樣長，唔止係你冇，而且係影響到家人嘅生命質素都冇。所以我問一個比較實際啲嘅問題就話，我哋好多同學仔喺度啦，有好多學者喺度啦，我哋會喺做咩嘢？下一步裏邊做啲咩研究或者係投資喺邊一方面，係會令到我哋可以改善呢個數字嘅嚇。
其實之前談都有好簡略咁提過。其實如果係講用證據嚟説話嘅話咧，咁我哋係好少有一啲證據係往前望做嘅，即係話我今天開始呢個研究，幾年之後比較另外一個 即係呢樣嘢少，但係我哋有好多回顧性嘅數據，發現其實有啲嘢我哋真係可以做嘅。頭先唔記得係咪啊？講過話要我哋個 exercise， 咁即係個腦嘅 exercise。In that sense 其實啱嘅，即係話我哋有幾樣嘢，我覺得而家確實個數目我唔記得啦，大概話有14個或者咁上緊嘅 factors， 有啲咧係冇得改變嘅，你個 age 你個年紀，你之前受過嘅教育，即係你60、70歲嘅時候，你冇得翻去我又讀多個博士出嚟㗎咁，即係有堆咧，我哋係冇得改變，有得改變嘅咧其實有嘅。基本上可以好簡單咁講就係，即係好沉氣嘅就係正常或者健康嘅生活 ，which 即係我會再講啦，就係身體嘅運動，即係話你 physical， 即係確實係你做運動，你有腦部或者精神上高或者智力上高有運動，即係唔係真係 exercise 啦，但係即係你要 keep up 嚇。然之後你要有你個五官，即係嗰個聽覺同視力，肯定就係應該仲係中大做，就係話如果你聽覺有問題，你容易啲會即係個智力會，即係呢個其實就係另外一個相關嘅就係你個社交，就係話呢個其實就係話你係咪少咗一啲？即係唔係比以前少咗，而係即係咩原因都好啦。你少呢啲咁嘅外來嘅正常嘅刺激或者活動。咁另外就係嗰啲心血管嘅因素啦，所以你有運動啦，你要控制你啲如果有糖尿啊或者膽固醇嗰啲咁其實即係如果你咁。可能我會漏咗1、2個未必好重要，但係即係都係叫做呢個年紀可以改變得到啲因素。咁但係基本上其實但問題就係恒心啊嘛。我頭先講啦，同我個老同學講，佢話你冇改喎，我話係上高呢一橛冇改到下邊呢橛點，即係係我哋中學嘅年紀嗰個樣啦。咁我唔係話要一定減到好瘦，好似後生咁。老人科醫生有一堆人話，你係要有少許嘅肥胖，係好少許啦，就話你有病嘅時候做一個 reserve，即係嗰啲係 minor points 啦，但係就話你 keep 一個身體一個機能好啲，因為其實我哋話認知退化或者認知障礙，我其中一樣嘢冇講到嘅就係、其實喺我哋已知嘅阿爾茲海默症裏邊。如果你睇嗰個病理咧，有1/3以上，即係話嗰啲人肯捐啲腦，即係譬如頭先我提過嗰、個修女嗰個研究，佢哋起碼有1/3以上係有第二啲嘅嘢喺個腦嗰度，即係話佢唔會係單係有阿爾茲海默症，咁即係你整體嘅生活、整體嘅病，所以我哋好難話因為對一般人嚟講，你其實唔係擔心你係咪有阿爾茲海默症，你係擔心嗰樣病之後影響到你嘅嘢。即係其實冇人會計，即係用我 scan 一次個腦，我睇下我唔正常嘅蛋白個 loading 有幾多。如果我個 loading 係唔正常，但係我係生活得好、食得好、瞓得好，我開心。喺嗰個頭先 call 嗰個 study 裏邊，有啲修女係有唔正常嘅蛋白，即係話 by definition 佢係阿爾茲海默症，但佢生活都好正常，即係 so call 好正常啦。即係你90歲或者80幾歲咁，你總唔可以同個18歲打都可以打得贏啊！即係 in a way 係佢可以好正常，所以即係其實仲有第二啲因素，我哋係唔知得到。That's why 點解我頭先其中一張 slide 話個 precision medicine 嗰度咧係好困難啫。 現實生活裏邊有太多 factors， 你每一個 factors 都要計落去個 model 嗰度咧，你個 model 其實好困難啊！咁即係呢啲其實應該就係要啲 tech 嘅人啊，或者即係佢哋要諗辦法幫手，即係特別係即係用 AI 嘅形式嘅時候，你要 reduce 嘅時候，你唔可以亂咁 reduce 佢，你唔可以斬咗堆話呢一堆嘢都係未必係用，即係呢個係要即係 that's why 我會話啊，跨學科嘅人一齊嚟去 aim at 嗰樣嘢就係你有啲生物醫學背景嘅人話邊幾堆嘢，你如果真係冇咁嘅資源嘅，你就 discount 嗰樣用 model 先啦。但係即呢啲就係要咁樣即係跨學科去做。咁重複簡單啲咁答翻你就係、其實可以肯定咁講嘅就係頭先嗰幾樣嘢係唔使食藥嘅治療。如果係用藥物治療嘅話咧，即係而家已經有就係話啊，如果你喺肯定你係有阿茲海默症，你個掃描裏邊見到係有嗰啲唔正常蛋白，你打嗰啲針，理論上係唔係理論上？實際上你一定會減少咗嗰啲唔正常蛋白。暫時我哋呢幾年嘅觀察裏邊，佢哋係會比唔打嗰啲人好啲。嗱，然後我講咗咁多嘢，其中我話暫時或者去到呢個階段，做咗5年嘅研究嘅喎，10年之後咧，15年之後咧，究竟佢係幫你好頭嗰3年，跟住即係話，即係譬如話，我等於我由83歲變翻80歲，咁但跟住嗰條 curve 都係一路係咁嘅，定還是我由83歲變咗由80歲，但我一路都係慢啲，我好過你唔打嗰啲人。我哋冇數據嘅，因為嗰樣嘢都係幾年㗎嘛。嗱，呢啲亦都係一個現實生活中點樣要去將現實嗰啲數據去擺落去，再做我哋下一步。所以我只能夠好確切咁話，有咩可以你而家做？就頭先講嗰啲酒唔係一個絕對唔可以，但係過量嘅酒啦，過量咧係冇定義嘅。唔係俾個機會你 escape， 而係話科學上高太多 studies， 每個人嘅過量咧係即係每個研究嘅過量係唔同，所以變咗你呢個參數你好難有個好客觀。但係煙咧就從來都冇喺即係 Dementia 呢度話食少少煙冇問題，即係呢個咧變咗就係煙係一個即係 yes and no 啊。
我諗頭先 Professor 話 AI 係因為 correlation 啊嘛，即係好多嘢係相關嘅，所以我哋例如 AI 都係計好多 multi-factor 嘅 model， 所以唔好 miss 一部分嘅 model 啦。咁我可唔可以話其實即係降維啦，即係將個 dimension 降低咧，就係話例如同朋友一大班朋友係星期六喺一個地方，咁然後研究啲咁專業嘅題目，呢個都冇腦嘅非常之好嘅一個訓練啦。
梗係啦，
就係讀翻個科大嘅 PhD Master 呢個世界級嘅，比 UCL 同哈佛更加高級嘅，同埋更加 effective 嘅一個啊。咁另外就可能同啲朋友去旅行啦，同老婆多啲去睇唔同嘅戲啦，唔好淨係睇愛情片啦，睇下恐怖片啦，咁你嘅腦特別受到唔同嘅刺激。但係咧，好多人咧都會有個 excuse 嘅，有個話你睇高錕，你睇中大嘅校長嚟嘅喎，佢咁聰明攞諾貝爾獎，佢成日都用腦啊，係嘛？點解成日用腦嘅人都會 dementia 咧？呢個係有冇咩原因？
呢個就係頭先即係呢個就係即係頭先講話，即係你一個複雜嘅病，多因素嘅病，你喺分一個即係你係成個 group 去做研究，即係成個 group 去問嗰條問題，同你某一個人你問呢條問題個分別就係話，我哋唔知佢係喺呢度定係喺呢度，我哋只可以話大部分係咁樣。即係如果我哋做研究，我哋將條 curve 成條 long 即係成個中型曲線往左往右近，你知道係有分別。但係你單一個人嚟講，你就唔可以話好 佢喺呢一度係有嘢定係冇嘢？即係你可以話，其實佢可能成個genetic loading係容易啲會出事，即係會有呢個病。我即係冇 inside information 啦，但係你公開嘅信息，佢家族係有人都有。咁即係換句話講，其實佢已經係話 preload 咗，佢 more likely 佢係企喺呢一邊啦，即係佢好容易就會過咗界。即係如果你用呢個簡單方法就係佢生出嚟，佢已經有幾粒細嘅細胞。你再多幾樣嘢，咁你就咁呢個就只能係咁嘅解釋嚇，即係唔係話一定
即係佢個 weighting 其中一個碼係好大嘅，話其他可能就少嘅，其實已經推遲咗咯。
係啦，即係佢可能已經係，即係佢有好，即係佢本身已經可能係好容易出事，但係因為佢係個前半生嘅時候係好積極，咁所以佢已經好咗啦。即係我哋冇辦法將一個人嘅歷史重新還，去話佢下一期另外一條 path 係咪差啲？即係呢個就係
冇 control experiment，冇一個
即係冇得係啦，冇因為你一個人嚟講，你冇得 control。
咁係咪我哋學新嘅嘢？都通常啲人話學新嘢啊嘛，即係發覺個雪櫃裏邊有條鎖匙揾嚟揾都揾唔到嗰啲就係 Dementia 嚇，就唔係即係咁我哋學新嘅嘢，其中一個係咪我哋專登學一啲我唔想本來唔想學嘅嘢？即係 against 我本來個 will， 其實呢個係因為其實某程度上好多人我成日覺得咧，例如我有基督徒啦，咁我成日每個禮拜都喺度查經啦。咁其實理論上好奇怪㗎嘛，查經你嗰幾行中文咁樣查成晚喎，幾個鐘成個鐘個幾鐘喎。但係你睇短視頻好快樂㗎嘛，幾秒鐘短微劇嗰啲好快樂㗎嘛。但係係咪頭先話修女啊點樣？其實有一個因素係你查經咧係透過幾個字咧喺度升維度啊，喺度 raise 個 dimensions。你喺度諗嗰個 generalize 或者係諗背後嗰個原理，嗰個過程裏邊其實都係除咗打麻將同埋打橋牌，其實查經都係。
應該咁講，我估即係呢個即係如果要靠用證據嚟説話，我估嚇就當然我應該好難 search 到，就係話係咪有啲乜嘢嘢即係認知上高嘅活動係冇幫助嘅咧？我估可能有一個研究去好確切咁話俾你聽，所以我即係整體嚟講，我會話大部分嘅應該都會 OK， 無論你話你查經或者做第啲嘢，但係我冇印象見過有人講話我特登揀啲我唔中意做嘅 task 或者嗱唔中意同 difficult 係兩樣嘢啊！即係你可以好中意，即係我睇嗰個我 prepare 呢堆嘢。最困難嗰部分其實就係中間 what is the substance of them， 因為嗰樣嘢我平日係唔需要做嘅，需要有即係但係嗰樣嘢我都係我中意，但係嗰日你頭先嗰條問題就話你揀啲你唔中意嘅，咁所以呢樣嘢咧，我覺得就係冇證據底下，我嘅建議係做你中意嘅嘢，不過都係要 challenge。但係你唔需要特登揀樣嘢真嘅，介意唔計嚇？
我問多一個可能，咁同呢個 BDNF 有冇關嘅？呢個 fertilizer 而家好興講啊嘛，個 BDNF 嗰個令到 dementia 可以舒緩嘅一個，今日冇提到啦，嗰個
BDNF 一定同 dementia 有關係，但係個 direction 係點咧 ？sorry， 我答唔到你，因為我冇做嗰啲 experiments。不過講開，即係突然間講開，即係你講，如果你講 environment 嘅話，咁頭先都有人問過帕金遜，就話 environment 其實有好多嘢係 affect 嘅，即係你即係我本來其中一個 joke 就係話，你想預防帕金遜症，千祈唔好去美軍基地附近，因為美軍基地咧有好多 flow compound，which is known to be 即係容易 causing Parkinson's 出晒㗎啦，出晒 publications 㗎啦，即係呢個唔係非機密嚟嘅，即係連美國自己嘅人都話即係嗰個 statistics 即係所以沖繩島嗰啲人係會慘啲咯咁即係 environment 係有關係，但係太多嘢有關係，變咗你好難有 individual factors 有
collective 嘅 reason。
呢度呢邊先，你哋舉咗手先。
Professor, thank you for excellent topic talk. 我諗喺 sorry 我咁講喺呢個 audience 裏邊，我諗我最 understand。我從事呢個生物醫學研究40幾年。咁其實我回翻，其實我 supervisor 喺 Stanford 嘅時候寫過嘅，一個好 science 咧係 answer few questions， 但係 waste more important questions。喺度教授過去做嘅 genetic studies very excellent， 因為即係呢個一個非常好重要嘅 foundation。但我講1950年代嘅時候，科學家就問究竟咩係 genetic material，DNA、RNA 定係 protein？當時1953年嘅 double helical model Watson and Crick，1956年嘅 Alpha Convert 我個 supervisor 就係 DNA polymerase 嘅 finding 同埋 purifies， 咁就1959年慶丁諾貝爾獎。然後1961年Watson and Crick 就拎諾貝爾獎。咁Stanford 嘅Paul 同埋 Stanley Cohen 就係 develop 咗 genetic engineering。咁1980年拎諾貝爾獎。你睇下成個 genetic study 教授所做嘅個 DNA mutation 嗰啲資料實非常 excellent questions。咁我其實喺20幾年前就喺長庚大學醫學院嗰時校長，嗰時我係research development 嘅，即係香港嘅副校長。咁校長係 engineering， 咁就叫我就set up 一個 genomic centre。咁我同醫院長庚醫院嘅 clinical professor 同醫學院嘅教授，我哋 develop 咗頭先所講 multi-omic， 就有 transcriptomic， 有 proteomic， 有 bioinformatics， 有 animal image centre， 有同埋嗰個image centre， 咁我做得非常好。咁我喺10幾年前我 sabbatical leave 嘅時候，我曾經有去喺哈佛兩個月，喺 Stanford 2個月咁我喺 Stanford 有俾 talk， 有哈佛俾 talk。咁喺 Stanford 嘅時候咧，有個 professor 喺哈佛去到 statistics department 做 head 嘅，非常有名嘅 statistics。咁佢就想做用 statistics， 當時佢用一個方法，因為細胞裏邊有2萬嘅基因，咁佢就做到見到一個基因 A， 見到基因 B 或者見到基因 C， 嗰所謂 co-expression、RNA co-expression。咁佢就問我可以點用，咁我就翻嚟用係因為我做 cancer study。但好有趣，因為見到 A、見到 B、見到 C， 突然之間見到 A 同 B 出現多咗好多嘅時候，就會睇 system biology 究竟佢係喺度分成乜嘢。咁我用呢個我就做咗好多 cancer study， 睇到好多好精彩嘅嘢。啱啱我頭先個問題問點樣去 prevent Alzheimer， 我就 come across 一篇文章係唔知哈佛定 MIT 做 Alzheimer， 就睇到幾個基因。係同 Nancy 佢哋咁多年做好多 Alzheimer 有關嘅字，佢哋睇到啲 gene 嘅 interaction 嚟到去解答頭先教授所講好多 gene study 呢咗好多重要嘅 foundation， 但係睇個 functional dynamic 嘅係睇到 what's going on， 咁嗰啲RNA 嘅 gene co-expression 其實係可以回答個問題。咁所以我教授覺得你後邊 weigh 咗好多好重要嘅 question。其實呢個 multi-omic transcriptomic 就係Alpha Convert 嘅仔Walter Convert，Walter Convert 就拎諾貝爾獎，幾時嚟香港嘅？喺呢個 Convert 都講。咁其實而家咁多年嘅 GWAS 嘅 study 咧，其實拎咗好多呢啲 genetic map， 但係後邊咧就係我哋啲 young scientist， 今日有好多 young scientist， 其實就係跟住我哋拎咗咁多 foundation 咧。嚟到去一齊嘅努力，最後我解答，其實我哋就係睇細胞個 homeostasis， 中文叫 homeostasis， 就2萬個 gene 係點樣去平衡。頭先教授有邊個個 balance 點樣平衡喺我哋運動喺我哋營養點樣去平衡我哋嘅生活嘅品質，我哋嘅 quality of life， 呢啲就係我哋咁多年大家教授努力嘅嚟到做嘅結果。咁我又 sell， 我最近寫一本書。係講 cancer 嘅，啱啱我會再傳過去俾大家顧，裏邊係完全講晒呢40-50年我喺醫學院嘅教學研究嚟到講出頭先我講嘅 homeostasis 點樣平衡，點樣住健康。最後講一句，好多 disease，Alzheimer 同 cancer 同心臟病同好多 disease 我就咩類風濕關節炎。頭先講大有個教授同埋廣義醫院醫生揾過我。 我哋傾點解用gene expression 嘅方法嚟到去幫類風濕關節炎，因為嗰個 disease 咧，原來醫生開藥開到唔單止開類固醇，有時係開啲抗癌藥，因為冇藥開啊。咁我哋研究睇到好多common feature 咧 ，Alzheimer 同 cancer 同同心臟病咧，其實好多 common feature 就係頭先我所講 homeostasis 嗰啲，我就
我哋嘅8篇學小朋友，我知道個個都有個證嘅，就係個麥克風敏感症嘅，揸住嗰個
有一個問題我啱新學㗎，就係原來咧，我聽過有個睇法就係一個健全嘅 cell 有個死咗嘅 cell physically 係 identical 嘅，咁咧但係咧就係究竟個生嘅 cell 比死嘅 cell 多咗啲乜嘢令佢有點樣嘅生命嘅本質係咩？有冇嘅聽你見解？我懷疑未知嘅。
我你第一個前提我係同意嘅，因為生同埋死咗嘅 cell 係有唔同嘅。你話生嘅 cell 有佢死亡由個 cell 由生到死亡嘅階段咧，都係有一段時間嘅。佢有一段時間咧，佢係基本上你係可以再驗證翻佢，即係你可以話佢冬眠咗。喺嗰個短個 stage 裏面係咪你見唔到分別咧？咁呢個我唔敢肯定，因為我畢竟唔做 cell 把嘅。咁但係你話生同死嘅 cell 咧，係每個階段都有個好明顯嘅分別，或者又係 cell 入邊嘅即係 suborganism， 即係佢再次細胞嘅粒體係應該會都慢慢。
咁我想 follow 問下呢個 homeostasis 同 serotonin 會唔會都有關比較？
你可以話 serotonin 係整個人嘅 homeostasis 嘅一部分，因為你 homeostasis 係一個好大嘅 concept， 咁你要睇下，即係
system level 嘅 concept。
即係你，但係你嗰個 homeostasis 你話我落到去個 cell 嗰度，或者我落到 cell 嘅時候 organ， 你都要有一個 balance， 即係譬如話你再落去喺度 conjure， 佢要 maintain 佢嗰個pH 啦、electric balance 啦，即係嗰堆全部都有，咁所以即係用最粗鄙嘅答嘅方法就係，serotonin有冇 involving homeostasis 就係媽媽係女人，即係
好大 component 嚟嘅。
但係即係呢個又一定會有，即係佢 acting 有，因為佢有 multiple pathway、multiple actions 喺個 brain 度，咁所以佢一定會有。
都同樣嘅 pathway 上冇講。
有，佢哋係同呢個
cortical 同 limbic 嘅都
試下。
凡係都會有 serotonin 咁 pathway 係有傳染，因為佢哋咁樣數嚟聽。
其實我哋想問，因為我唔識㗎，layman 嚟嘅。我個 roommate 咧就做老人科嘅喺美國嘅，咁佢就話咧即係而家講緊嗰啲咩老人痴呆咧驗就好易驗，但係而家冇人肯做嘅喺美國，因為驗咗之後咧發覺冇得醫嘅。咁我想問一個好 stupid 嘅 question 咧，就我哋而家換心啊乜都可以換，個腦可唔可以換嘅？
換咗係咪你自己嘅？
所以就係即係其實都答咗啦，即係個腦第一個假設，即係你有辦法買到個後生仔、後生女嘅身體，你係咪真係可以換㗎？你換嘅時候，即係我用個好科學嘅解釋就係，你換幾多嘢？你係你喺個頸上都斬咗。對唔住，咁你以後就唔行得㗎咯喎，你會唔會？你話 OK， 我用啲我成條 spinal cord， 即係成條脊椎我換晒 OK， 你去到 spinal cord， 但係 spinal cord 出嚟嗰啲即係你個 concept 就係話，你就算假設佢成功，你換嘅目的係乜嘢？ 你想長遠長咗30年、50年命，但係你付出嘅代價係咩？咁所以你話係咪可以換？你永遠都可以做一個咁嘅 experiment 嘅，我哋喺某啲地方，但係 technically 另外一樣就係你 technically 你換咧，你其實你要縫合同埋 link up 好多嘢，咁嗰樣嘢，well 起碼我覺得眼前10年我會覺得呢個係一個 feasible 嘅，即係一路都會有人做呢啲咁嘅 test。
係好多謝教授嘅意見。咁我公司就係主要做人形機械人嘅，喺香港做嘅。咁我開發咗一個新嘅 product 咧，就係咧將之前咧外國有個 research 咧係將呢個 digitalized brains 將嗰個果形啦，嗰個腦咧切片嘅，咁就將佢嗰個腦咧直情 digitalize 擺落去嗰個電腦上面温嘅。咁我公司就將而家就再進一步，就將嗰個果形咧，就將個機械人結合咗。咁而家就可以將嗰隻機械人咧就可以嚇嘅。其實一隻係嗰隻好似蜘蛛咁嘅，咁我就 modify 咗佢，用 Nvidia 嗰個Jackson 啦，咁佢自動可以嚇嘅。咁佢就做埋嗰啲有 sensor 有 eye sensor 有smell sensor 有 taste sensor 咁。咁我發覺一個好得意嘅嘢嘅，咁就係我發覺兩隻機械人咧，都用同一個腦，即係嗰個外國嗰個open source 嗰個腦啦，個電子腦啦。但係我發覺佢哋兩個腦咧，對兩隻機械人咧，嗰個嘅興趣咧係唔同㗎。係啊，一隻咧會係即係一樣㗎。出咗嚟之後咧，佢哋一隻咧會中意鹹中意甜啲嘅嘢嘅。咁一隻咧就會中意咧冇咁甜嘅嘢嘅，咁有冇得解釋得到？暫時好似我睇到出邊都未有解釋得到㗎啦。
呢個我完全即係冇 touch 過啦。不過我反而好有興趣。你意思係話佢將果形嗰個腦，將佢成個 model 擺咗落去一個 computing 嘅 system？ 係
啊，變嗰個起上咗 Nature 嗰個嘅 journal 㗎啦。咁我再進一步係將直情隻嗰個果形嗰個腦啦。係擺落個機械上面，擺落嗰個Nvidia 直情。將一個 log 擺咗入去。係啦，係擺咗落去嘅。咁佢而家就全世界第一個嚟嘅，咁我就做得到係擺落機械度。咁我哋仲出埋 SDK 可以俾賣㗎啦，開始揾大學合作。但係
呢個我覺得就等於你我自己呢個就係我一個作為一個非專業嘅理解下，因為呢個唔係我嘅即係。我會理解就係你等於一個 AI machine 一個佢喺一個從個開始嘅 stage， 你喺唔同嘅 environment 裏邊，你 train 佢嘅嘢嘅時候，佢自己都會有唔同嘅。
其實數咗幾張㗎，
即係我咁你如果係將兩隻果形，你係 assume 你就上有一個 gene 嘅 background。即係話你升，即係將佢一個 cell 拆開兩個，然之後調一兩，即係所謂我哋嘅孖生兄弟。我哋嗰啲孖生人類都有唔同嘅性格，即係所以我覺得係 other than error 之外，我覺得佢嘅 learning 嗰度會 force 相嘅，即係所謂嘅，即係從一個生物嘅角度去估去解釋你嗰樣嘢，但係呢個係有。
教授，我想問下就係過去咧人生老病死大家都知㗎啦，係即係同 tax 一樣都冇法律避免嘅。而家做説法咧 David Sinclair 咧係呢個 Harvard 嘅 medical school 嗰度咧，佢認為咧即係呢個係一個 disease 嚟 ，aging 係一個 disease 嚟嘅 ，can be cure 我成台呢度咧都唔少人做cell therapy 嘅。stem cell 咁都係即係似乎有一定嘅功效啊，講緊其實目的就係延遲嗰個衰老咁當然我哋都做 exercise 所以我哋都做啦。但係我哋咁樣做晒啲嘢，我哋都死㗎嘛，要做係咪？但係而家説法就唔係，而家説法咧個咧就係longevity escape velocity， 即係話咧只要我哋活得夠長多少少咁咧，因為個科技發展好快，係咪？即係以前我都冇聽過，即係UFO 啲花唔得開啦，係咪？ 我哋好多嘢都冇想象過，但係近呢10年8載係好大突破，係嘛？我哋講有 AI 可以 break down 好多嘅 protein 佢哋個 structure， 呢啲咁嘅係劃時代。嗱，講緊係好短時間之內發生，以前係好耐，而家唔係，講緊一個 generation。個問題就係呢個咁樣嘅 logic 係咪可以有機會令到我哋長生不老？嗱，呢個都嗱即係短嘅説法，你聽到習主席啊、普京佢哋都講緊120、150有咩問題啊？咁係咪有機會即係去到長生不老或者 substantially increase 我哋嘅歲數？我想問呢個問題，咩啊？
呢個即係亦都係冇證據嘅估計啦。不過我盡量用我自己嘅諗法去，我覺得呢個係即係可以話啱嘅，即係個方向係啱嘅。即係你如果呢樣嘢都保持好 optimal， 你係可以長壽多好多。但我覺得唔會係一個所謂嘅長生不老，因為第一，如果你講 regeneration， 你喺邊一個 age 開始 start 你嘅 regeneration 啊？即係我所有心肺等等所有嘢我都唔講，只係講個腦啫嚇。即係假設嚇，你係假設個腦係完全冇一個衰老，而你嗰個係或者你係有衰老，但係你係完全可以改善得到。咁我覺得眼前我唔認為可能，我唔敢講話20年、30年、10年，我覺得我個 estimate 都唔可能。原因就係一，除非你而家仲係好後生，即係話我啲我自己睇我自己就話，即係我即係有時講笑就話，如果我要有嗰啲 abnormal protein， 即係譬如 amyloid deposit 嘅話，咁我開 deposit 可能都 deposit 咗啊，即係問題就係有 last 幾耐，即係話你個 intervention 嘅時間，你要介入嘅時間，唔係講緊50歲、60歲啊，我覺得係要早好多啊，因為again 我唔記得咗邊一篇 paper 啦，佢哋有人計過，即係唔知點計到出嚟，個 brain cell 嗰個 cell number 20幾歲20到蚊就已經開始跌嘅，咁你 at that stage 啊，咁所以另外就係話你 support 嘅時候，你個 brain 你要 support 個 brain， 你要有好多第二啲嘅 supporting factors。而係咪每一個 factors 你都可以，即係每一個因素你都可以搞得到？嗱 ，mitochondria 係其中一個好重要嘅嘢，因為 mitochondria 佢好其中一個重要嘅部分嘅嗰啲遺傳因素係一個 mitochondria， 即係話你係而一個 cell 裏邊有即係幾十到過千視乎佢需要啊。咁每一個單位。佢都係有佢自己嘅 production， 即係自己嘅，即係話你假設有1000隻mitochondria，你第一隻可能一路 function， 第二隻佢唔知係邊個 stage 佢開始係 normal， 但佢係一路 accumulate 入去，佢可能將呢個 abnormality 一路再 pass 落去，即係話你要諗下，其中有即係你要控制嘅因素太多，多到我唔覺得。現階段你係講緊可以話長生，即係甚至唔講長生嘅，即係你講 another 100 or 20 years， 因為 another 10 years 咧，我覺得哇，你 keep 好多嘢，你已經可以改變得到嗰個 range。咁呢個就係其中一樣就係我哋而家好多 therapy 啊 gene therapy， 其中有一啲嘢就係你去到某個階段，你先再改嘅時候，你可能係要 target 好多個 cell。咁而等於你呢個就係、你點樣可以 target 咧？得好少嘅嘢係一個 good gene therapy，blood disease 啦。咁你因為你改咗少少，佢慢慢自己 reproduce。眼有啲你 infuse 入去，你嗰個眼底好多都可以直接。其他 muscle， 除非你要有一個好嘅 vehicle， 好 specific 去某啲 cell。 而呢啲 cell 係適合做一個 gene therapy。咁所以我覺得我聽過知道有好多人係追求，特別係嗰啲 super rich， 但係我覺得120係feasible。但係再耐啲，我覺得難咯。但係佢哋嗰個 age group 可能佢哋講120去到100，could be possible。
所以大家星期6日或者假期上去深圳去按摩嘅時候，唔好記得除咗享受按摩之外，仲要massage 住個腦啊！即係幫自己嘅腦 massage，唔係就你個身體 massage。我諗時間差唔多，同埋但係咧，我哋今日其實有今日嘅咁精彩嘅。我成日覺得係 inward 啊，即係大部分人覺得學嘢咧都係學 outward 嘅，即係學工具點樣用嘅。但係咧係唯一一次百川學術咧係 inward 嘅，即係我哋研究我哋自己嘅，研究我哋自己永遠都係個智慧提升最多嘅地方嚟嘅。帕斯卡爾講啊嘛，即係好似蘆葦草啊嘛，個蘆葦草其實好容易俾啲風、俾啲咩雨啊就整死咗。但係呢個蘆葦草有 consciousness 啊，有嗰個有個 consciousness 有個意識啊，而呢個意識係仰望星空嘅時候，唔驚呢個宇宙嘅力量嘅，佢係會想知道多啲關於呢個宇宙嘅事，但係宇宙對呢條蘆葦草可能反而係一無所知。咁我哋今日非常之感謝百川學術啦，Wallace Stephen阿關同埋最緊要多謝Albert，Professor Albert 即係將科大嘅呢個講堂帶到嚟呢度呢個，因為Professor Wong 應該好少，應該我喺網上面好少見到關於佢嘅，好低調嘅。其實佢唔係成日演講嘅應該，但係學術嘅演講好多嘅，即係嗰啲 nature 啊、啲 science 嗰啲演講好多嘅。佢光圍同我哋講呢個係好少見嘅應該係，所以我哋非常之感激佢即係係咯，同我哋講得咁精彩嘅 enlighten 嘅呢個。
多謝阿 Ben 啦，多謝。咁留一留步，我哋咧就好環保嘅，我哋 earth friendly 嘅，所以咧我哋就冇呢啲咧水晶膠啊嗰啲咁嘅紀念品嘅。不過我哋會有幅相啊，阿 Wallace、Professor Yip、Stephen Wong，我哋主要啲即係我哋大義工嚟嘅，我哋自己都要俾錢食飯嘅嚇，我哋即係所以係百川咧係人人平等，亦都咧做到咧，我哋希望能夠咧繼續持續有個 sustainability， 可以有唔同嘅 topic、唔同嘅課題可以嚟到咧，大家咧即係獲益良多。好啊，我哋先上先，應該短時間咧就先。
補充一句，呢個無論 NVIDIA 股價幾高，呢個 Scinece 幾勁，其實我哋嘅圖係最勁嘅。
望住呢度先。
So far 都係，我要敬畏呢個創造者。
咁多張啊！1、2、3，用呢部機再影啊！1、2、3，
好，請就座，請就座。咁我哋坐定定咧，我哋咧就稍後可以嘅，會嘅。咁我哋坐定定咧，我哋有一張大全場嘅合照。